Okay, so I'm back and let's start discussion again.
Excuse me, sir.
Yeah,
Sir, how does the first DNTP interacts with the DNA template?
the first thing that you are saying, right?
Yes, sir.
Yeah, good question. So you will get to know this thing like within few minutes. Yeah, good question. So here DNA is double helical, hence the two strands has to be separated from each other. How it happens? Inside body we cannot increase the temperature. So what happened here in this part you can see that double stranded structure. We are not showing the helix yet. Double stranded structure. But still this double stranded, that means this hydrogen bond needs to be broken so that single strands are available for the synthesis. Like as far as I explained now, that you need a single strand that is using as template so that whatever information written in this strand, it will copy the other strand. So that is semi-conservative replication. But supercoiled DNA relaxed by topoisomerase, and so here we are not showing the supercoiled DNA yet. I will show you right now. And then this hydrogen bond that can be broken by this enzyme called helicase. So this is an enzyme. So that breaks this hydrogen bond. So that in room temperature, this hydrogen bond will be broken so that it can be single stranded. DNA can be single stranded. And then the synthesis will start, right? Then synthesis will start. So now the question is, what is supercoiling and how topoisomerase actually helps? Topoisomerase is one enzyme name, all AACs end with AAC, that is some kind of enzyme I told you before. So now uh, I will actually show you here. See, in DNA, like in E. coli, you have one circular DNA like this. Okay, this is a circular DNA. So you have like two strands, they are actually making an helix, and this is circle like that. In E. coli cell, you have one circle. And now forget about this circle. With circle, it is more problem. But in our body, we have linear DNA like this. But inside one cell, we have 46 DNA molecule. Inside one cell, we have 46 DNA molecule like this. Now, since DNA are double helix, like they can form the helix, not just double stranded. So as a result of that, what do you think? That helix, like two strands are here. Now, if you try to open this two strand like this, see the other part is actually moving, rotating. And DNA, some DNA are very long, whatever present in our cell. Some just one DNA molecule is 2 meters long, like the longer one. And you know the dimension of DNA that per one turn you have how many nucleotide pair. So if you do like this, so that will create like, this will like the end part is like moving like this. It will create turbulence inside the cell. That is not going to happen. And in case of E. coli DNA, it is more problem because it's a circle. How you can actually pull this thing? So everything will be tangled together. So this is not going to happen, right? This is the problem of this helix. So this issue needs to be solved. So how you can solve this? If you have two strands like this, unfortunately I get this rope where the three thread together actually. So ignore that. So you have just two. So here you have two thread, but
Sir,
ignore that.
I have a doubt.
Yeah.
Sir, here we are separating the three threads with force. That is why the turbulence is being created. But in the case of DNA molecule,
Hmm.
the helicase enzyme breaks the hydrogen bonds, and they would just separate. There would not be any force like we are applying in this. So why would there be a turbulence in that case?
Yeah, but DNA will like that is already helix, that is coil, right? If you like open the if you open the hydrogen bond will be broken. But the thing is how you will release the helix. Like if you just think like even after the class, you just make one rope using like 2 thread, right? Now, how you can separate those 2 strands? Because if you are, I'm telling you, okay, you are breaking the hydrogen bond, but in order to separate those things, then you will, if you try to separate this, then you will put lot of constant here. This part will like rotate like this, and if it is equine circular DNA, then you cannot do it. Tanmay, did you get it? Because
Yes,
this
sir.
is,
Yes, sir.
yeah, so that is the problem. So in order to like remove this problem, what happened again, I am telling you that thing that those two are like 2 strands here. This is called replication fork. In that slide you will get something like this. I hope you can see this. So now these are the 2 strands. So in our body, in our cell, what happened very cleverly, one enzyme is present. So just when helicase is trying to break the hydrogen bond, the other enzyme, and that is called topoisomerase, that actually clip one strand here, then release this constraint. That means clip one strand and release this constraint and then again sell it. So that way it actually solved this problem. And now see, the enzyme is called topoisomerase. The name is topoisomerase. You will never forget if I tell you why it is topoisomerase. This DNA molecule, whatever the molecular weight, if you separate this, like whether it is like helix or it is double stranded, no helix, their molecular weight, everything will be same. They are just isomer. What kind of isomer? Topological isomer. The 2 stranded, double stranded DNA, but you made an helix out of it. So those 2 are topological isomer. They have some topological differences. And the enzyme that actually altered this topology, that means from the helix to double stranded, so that is called topoisomerase. That's why the enzyme is called topoisomerase, right? So again, coming to this slide. So here the supercoiled DNA is getting relaxed. That means the coiling is getting relaxed by topoisomerase, the enzyme, and unwound by helicase. So another enzyme that breaks the hydrogen bond. So as a result of that, you will get this fork. So this is called replication fork. So you can extend DNA in this direction long, like you have long DNA here. Just imagine you have long DNA here, like maybe another 5,000 nucleotide long. It can be even more. But you have to first generate this fork so that you are getting the single strand in order to synthesize the new DNA. So this is the direction of movement of replication fork. So as a result of that, what you can expect? So DNA will be synthesized here, and then again this fork will move in this direction. So that way, let's see what are the solution here. So helicase, one enzyme that needs some ATP. This is just for energy, but finally they will break the hydrogen bond. So together, topoisomerase and helicase, they together work to solve this problem, and that's why even in ambient temperature, in our body temperature, this can happen. And then some protein, those are called SSB. S stands for single strand binding protein. Because you might ask me now, like some student, like you might ask me that you are separating those 2 strands, right? But this is room temperature, not high temperature. Again, those 2 strands will fall up, like fall together, like fall into place, I would say. So you separated these 2 strands like this, but it's room temperature. We are not increasing the temperature. So again, it can anneal like this. In order to prevent that, some proteins are present that is called single strand binding protein, SSB, that actually bind to DNA so that again it cannot form the double stranded structure, so that it is allowing some time for the synthesis of the DNA. Okay, so that's the idea. So, but that is,
So
I'm
I
not.
didn't understand how the SSB is preventing the combination of the 2 strands.
Yeah. So see this, in room temperature, if you separate these 2 strands, again it might come together and it form double stranded structure, right? But SSB or single strand binding protein, they interact with the DNA or single strand DNA because their substrate is single strand DNA. That's why single strand binding protein. So if like please mute yourself. Yeah. So as a result of that, if it is getting attached to the single strand DNA, so that is preventing to form hydrogen bonding with the another strand. Right. In the figure, we are showing like this that those two strands are separated and like this. But in actual case, those two strands are still very close by. They might form again double-stranded structure. In order to prevent that, for some time during the replication process, this protein binds and that prevent the formation of double strand again during that time. Okay.
Sir, are they sterically hindering the formation of hydrogen bonds, or are they themselves forming hydrogen bonds with the
No,
fluid?
no, no, no. They just block. They just block their those nitrogenous bases. Okay, so that
Okay.
they cannot form hydrogen bonding. Yeah. So
So means when the single strand is formed, only then these proteins bind to the single strand.
right,
Not
right,
they are
right.
not bound before.
Double-stranded DNA. This is single-strand binding protein. This is, I would say, minor, right? So this is not like I would say major focus of replication. I would say just think in this way that we are carrying out this thing in room temperature. So there is always chance that again this double strand will like separated strand will make double strand because at this temperature they will try to make the hydrogen bonding right between those bases. But in order to prevent that for some time, allowing some time to carry out the replication, right? So these processes are very much dynamic. Actually, inside the cell, this process is happening very fast. Okay, only we are explaining very slowly because of this is a complex process, but this is dynamic and very fast process. So let me go through it. Then we will we will go through one video also. I think that will clear some of your doubt as well. So uh, now the second question is that first question I already discussed, right? That how it can be single stranded and how you will remove that supercoiling constraint. The second problem is DNA polymerase needs a free 3' OH of a pre-existing nucleotide to extend the chain. One student already asked me a few minutes back that how the first nucleotide will be added. This is very difficult question to answer. Like answer means to explain. DNA polymerase needs a free 3' OH of a pre-existing nucleotide to extend the chain, right? But then from where you will get the pre-existing nucleotide chain, right? So here the chemistry, like the reaction is simple here, that this is the existing DNA strand, and then from this existing DNA strand, you have the 3' OH group, and it carry out that nucleophilic reaction, and the next nucleotide added here. But
Sir, can you please repeat?
yeah, the question is that you need a existing nucleotide present so that it can if you have the 3' OH here, so that next nucleotide can be attached. Say I'm telling you here. So this is the template 3', 5'. So the new strand will be synthesized here 5' to 3'. As far as I discussed before and here also in this slide, see we mentioned here already that you have ACT, like short nucleotide segment, like short DNA molecule. And now in this T, in this nucleotide, you have this 3' OH group here. So it can carry out the reaction, the next nucleotide will be added. And whatever the sequence present in the parent strand, based on that, that will be selected. But the question is that how you will get this 3' OH here, how you will get this short segment here, because you have to start, but how you will it will be started, because here we are showing the short segment here, but even how this short segment has been started, because DNA polymerase needs a free 3' OH, so the first nucleotide can be added, we don't know the mechanism yet, right? So that's why I believe that some student already asked this question before. So now, in order to answer that, what I want to say, there's some complexities coming here. This slide is again the previous one. The way we made this slide is whatever one problem we are discussing, in addition to that problem, we are bringing the next problem. So this problem already solved how you can get the single strand. But now the problem is how you will get the the initial part of the DNA so that you will get the 3' OH group and you can continue it. So the idea is you make some kind of primer. Primer means, I believe that I need whiteboard, otherwise it will not be clear. So until now, what we discuss is so this is the replication fork here, right? This is replication. Fork. Now, the question is that DNA will be synthesized in this direction, but first nucleotide, like how it will be selected and how we will get 3' ways, because DNA polymerase needs 3' ways. So what happened during this synthesis, before DNA synthesis, little bit of small RNA will be synthesized first. Like this is weird, I would say, explanation, like completely new to you. I will repeatedly tell you again. So at the initial position, like a small segment of RNA is being synthesized. And this RNA or DNA, everything grow from 5' to 3' direction. RNA primer, right? So why it is primer? Why it is called primer? Say for example, even when you color your house, right, your apartment, before actual color you put some primer, right? Similarly, in this case, what happened? Like these are primer because we are trying to make, or we are trying to prepare, we are trying to replicate DNA, but initially we are making small portion of RNA, and this RNA will be synthesized by 1 minute. Somehow it get zoomed. Okay, so now
I am not able to understand how is the primer formation location specific. Like this could have been formed anywhere in the middle also.
that is possible. I will come into that also. That is also possible, right? But here the biological system is very complex, right? You are right. You can tell that here it can also happen, right? You are right. But only thing, DNA is very long, but only that this portion, like small portion that form this fork, right? This small portion right here. 1 minute. Because yeah, so only this portion is available. This is hardly, like I would say, 30 nucleotides here. It's unopened here. So as a result of that, not the whole thing is available, but it will go in this direction. When the DNA will be synthesized here, then the fork will move in this direction, and this portion already will form double helical structure. Like, let me go through a little bit, then I think you will be more clear. So now when the primer is being synthesized, and this is RNA, not DNA, and the enzyme called primase, because primer is formed by primase. The beauty of this enzyme is it can randomly add few nucleotide here. So, and it doesn't require the existing 3' OH group. So this enzyme, they can add few nucleotides here, just complementary to this strand, they will make some RNA. But finally this RNA will be removed. So you don't that doesn't matter what kind of nucleotide sequence added here. 1 minute, because one of my research scholar calling actually again and again. Just a minute. Hello, Ramda. বলছি হ্যাঁ নমতা আমি বুঝতে পেরেছি, তোমার সঙ্গে ডিটেলসে কথা বলবো। হ্যাঁ আমি অ্যাকচুয়ালি এটা ঠিক আছে। হ্যাঁ বুঝলি, আমি না ওই সেই ফার্স্ট ইয়ারের ক্লাসটাতে আছি, তো পরে কথা বলবো তোমার সাথে। ওকে, বাই বাই। Sorry, because this was really a critical problem actually. So one of my scholar facing. So that's why I just picked up the call. So anyway, um, so here, so what happened that like RNA primer, which is catalyzed by primase enzyme, so that will like make this RNA primer. So as a result of that, you will get this 3' OH group here, and then the polymerase, that is DNA polymerase. Here, what is mentioned here, polymerase 3. This is nothing but DNA polymerase. Then DNA polymerase will get that 3' OH in the existing RNA primer, and it will catalyze the DNA synthesis. Like 5' to 3' direction, it will make the DNA. So when the DNA is getting synthesized here, then again this part will be unopened. So it will be continuously being synthesized by DNA polymerase, right? So it will continuously being synthesized. This is called leading strand. Leading strand. So it leads the process. So some of those things, I'm sure that some of the questions are coming to your mind. Just bear with me and I will ask you, like I will give you time to address those things. So now the point is, what will happen into this strand, the other strand, how it will be synthesized? Because you cannot start like, so if you again, like if I go back, so here I told you that leading strand will be synthesized in this way. So it is being synthesized like continuously like this, 5' to 3', and again DNA getting double-stranded DNA here after synthesis. So it will go like this, and again the fork will move in this direction, it will start. But remember that DNA, RNA, everything grow in 5' to 3' direction. So as a result of that, it cannot grow like this because this is 5' and DNA is anti-parallel. So how it will grow? It cannot grow in this direction. Any solution? Can you suggest any solution, any students?
So first, small pieces will be added, then they will be joined later.
Yeah, exactly. Very good. So this portion is opened only. So in this strand, this is called lagging strand synthesis. It go a little bit slowly. So this portion is just open. This that makes the fork. So what happened? First, one small primer will be added here. Okay. So this is the 5' and 3'. And then one, like I would say, one DNA polymerase that will make DNA here, forming DNA. And then again, when this part will be like this part will be, unwounded. So another like RNA primer will be added here and it will be continued. So as a result of that, if you can imagine that when this fork was formed, so initially, so it formed some, small RNA primer here, and then DNA polymerase actually extended this. And then again, another primer here, and then again one DNA polymerase moved like this. So this is 3' and this is 5'. Okay, so that way it is going. So the lagging strand, small section synthesis. I hope you understand. The point is that the DNA is not fully open, that it is not a problem if at the initial point, if the DNA is completely open like this, like both strands are open. So you have 5' to 3' and 3' to 5'. Okay, so as a result of that, what will happen? So this strand will be synthesized like this, continuously it will be synthesized, and this strand will be synthesized completely like 5' to 3'. But it is not going to happen here. This is happening where? Inside the tube, when we increase the temperature, the whole thing get denatured, right? Not denatured, like they can unopened. But in this case, the problem is we are dealing with the small portion, right? So as a result of that, this portion is not yet available so that the DNA can be synthesized from here. So as a result of that, small segment synthesis happen. So now, if I go back here, it
So
will,
one question. So
yeah.
the primase always starts the replication from 5' to 3' only then.
Yeah. Any, like I would say in the world, nobody even discovered until now any polymerase like nucleic acid polymerase, like DNA or RNA, that can do the reverse way. 3' to 5'. That then that will solve the problem. Any of this, all of this enzyme, whether they are synthesizing DNA or RNA, that is happening from 5' to 3' direction. Why? Because of the availability of 3' OH group in the 3' end. Right? The 5' end is already capped with phosphate. So you don't have that nucleophilic interest, like reaction cannot happen there. That's the reason. So now I told you, so before that this process is complex process. I'm trying like as much as possible. If you can withstand this, then later part will be easy, because many students sometimes they lost like here like part of the like thing, but it should be simple after that. Like when we will discuss polymerase chain reaction, that is much simpler. So now if you like come here, so RNA primer for leading strand, you just need one RNA primer because it is continuously being synthesized. But in case of lagging strand, like when the fork was here, then one prime is added, one primer, this portion is being synthesized. And then another primer will be synthesized here. Again, it will be added like this, see? And then again another one like this, and then this portion is not yet open, so when it will go, and again it will be one prime is will add primer, and then it will be synthesized. So that way small segments are being synthesized in case of lagging strand. So these small segments of DNA is called Okazaki fragments, because it is a scientist name actually. Okay, you don't need to concentrate on that because it is not anything understanding here. So I think some Japanese scientist, he actually showed this thing, because the basic concept was discovered by Kornberg, but small you can understand there are a lot of problem actually solved by many other scientists. So this is called Okazaki fragment. And now what will happen? The next question is RNA primer need to be removed. That means we wanted to make DNA. We made the DNA, but unfortunately that DNA contains some RNA segment. In leading strand, you have just one RNA segment, but in lagging strand, you have multiple RNA segment in between. So we have to take care of this thing. Like somehow we have to remove this. Then we will get the DNA only. So the problem.
Excuse me, sir.
Yeah.
Sir, for the RNA primer to add to the first position, is it due to the property of SSB protein?
Property of RNA, property of
SSB
that
protein means the ones.
Yeah.
Means the
Property
ones
of,
binding to the like single strand. Is it due to their property?
Yeah, so primase actually like DNA polymerase, they need the 3' OH initially, but primase they randomly add some nucleotide there. So as a result of that, whenever they randomly add some nucleotide, then that nucleotide they are exposing the 3' OH group. So that's why it helps. But it is like not the permanent thing. This is some kind of solution. As I told you before, that primer is not the permanent solution when you are coloring your apartment. Right. Similarly, that primer is nothing like, they are not even complementary. They can be something else also. But the good thing is, like it will provide the 3' OH group so that DNA polymerase can actually synthesize the DNA. But this is the property of primase that can randomly start the initial part. Okay. And now, if you see
No,
here.
sir, I am asking a different
Yeah.
question. I am
Yeah.
asking that when the DNA is opened, means it's made in single strand. So for RNA primer to add to the only at the first position, is it due to SSB protein property like some steric hindrance or?
I don't know actually. Even I didn't get, because that SSB protein is bound almost everywhere wherever the single strand is there. Okay, so this is not that way, because like that RNA primase should add the primer, and then it can be, I would say, 2 nucleotide, 3 nucleotide up and down. That is how that mechanism is controlled. I believe that some mechanism should be there, but that is not like, we are not aware about this until now, right? So, and SSB protein is not like, I can read a little bit more about this thing, but I don't think that SSB has any role here in this case. Okay. So
Sir,
now,
does
Okay, sir.
SSB add on both? Hello, sir.
yeah.
So does SSB add on both strands or only on
No, both stand. We are just showing there. SSB should be very small protein. That is just, some of the SSB if bound to the single strand, then that will like prevent like those two strand to again, come together. That's all. And then when this synthesis will be happen, I'm sure that the affinity of DNA polymer is even stronger. So that will displace this SSB protein and the double-stranded nucleic acid will be synthesized. Okay, so let me go through the video portion. Then I think it will be more clear. Let me go through that actually. And I'm sure that I know that this is a problem. And every time when we discuss that, student they have a lot of question also. That is normal actually. The only thing is we are teaching in online mode. If I can use a board like big blackboard, that would be much easier. But the thing is, here the RNA primer needs to be removed because we are not interested to keep RNA inside the DNA because RNA is unstable, all of you know. So as a result of that, there is one enzyme present in our body, in our cell, that has the ability to like clip or replace this, RNA primer because of their some enzymatic activity that is called RNase H activity. So that will clip the RNA primers here. So if you forget this part, like the name of the enzyme, it should be fine. So you have to remove this RNA which are present inside the DNA. And now this RNA is removed by this enzyme. And now this DNA is already here. So this DNA has 3' OH group here. So as a result of that, now what will happen? DNA polymerase will actually continue this like DNA synthesis, and then finally the DNA polymerase will synthesize, but the last phosphodiester bond, that gap will be sealed by another enzyme called ligase. So DNA ligase. That's all. No more name, no more enzyme name, everything fine. So now I will show you just on video because then you will ask me question. I'm sure that you have question, but let me show you the video. That will make some some more thing clear. Then I will discuss again. So open the video first. I will upload this video in MS Team also. So can you see the slide video or not?
Yes, sir. I can.
You can see, right?
Yes, sir.
Because it's kind of I am presenting. So 1 minute. So can you see a DNA in a movie?
Yes, sir.
Yes, sir.
Yes, sir.
Okay.
Sir, is there any sound?
Yeah, sound is there, but you cannot listen.
No, sir.
Okay.
Sir, we have to
So
share the
1 minute. So it's fine. So I will actually narrate this thing. So the thing is here you can see that this is a double helical structure here. So here you can see that structure here. Now this is like this pairing. That means you have like T and you have the A in the other stand.
Sir, your voice is
See here.
cracking.
sir, here
Your
voice
voice is breaking.
Voice is breaking.
See, like strands are running in anti-parallel that I always told you that one strand will go like 5' to 3' bits. The other strand will
sir,
grow
the
like
voice
Sir,
is
we
breaking.
cannot
Sir,
understand
your
you.
voice is not coming
3'.
clearly.
So as a result of that, now
Sir,
Hello, sir.
Hello, your voice.
yes,
sir, we cannot understand you. Your voice is breaking a lot.
okay,
We still cannot understand, sir.
yeah, I understand. Maybe because of video actually. So what we can do? So I can actually just, load this video in MS Team and you can just go through it. Okay, so that will be better. I believe that everything like, video is streaming and I am talking. So that way it is problem. So now you can hear me well.
Yes, I think that was
Yes,
because of
sir.
the video sharing bandwidth
Yeah,
problem.
that is the problem because see, like in normal class, I used to show this video, but here the problem, I understand the problem. So I will upload this and you will just go through it. Then it should be fine. Everything should be fine. No problem. So if you have like, very quick question, you can ask me. Then I will move to the next part.
Sir, I have a doubt. Sir,
Yeah.
in the primer portion, after the polymerase extends the chain,
Hmm.
so the replicate means the strands have been separated, and the means antiprimer strand keeps on adding simultaneously. That's what you are trying to explain here. Means the two strands separate, and at the same time there is means ETCG and the corresponding basis bond at the same time.
Hmm.
So that's my doubt. Means is that happening, or there is some other function of primase?
No, primase is nothing. Just the primase is giving the primer. That means 8 to 10 nucleotide long small segment of RNA. Okay, so like I should like come into here. So here, like small segment of RNA that providing 3' OH group so that DNA polymerase can start from here. Whenever it is done, that one enzyme that remove this RNA primer, all this RNA primer. Okay, but if you remove this RNA primer, but still this DNA, it has now the OH group here. So as a result of that, DNA polymerase will actually make the DNA again here. And finally, one enzyme is present that is called ligase that will fill the gap actually. Gap means that one phosphodiester bond that needs to be filled, right? So. As I told you that that's why I wanted to show you the video, but this is not working in this way. So I will right now after the class within few minutes I will upload the video actually. Then see this video that will make things even clear actually. Very wonderful video actually. So I will do that soon.
Sir, in the upper part, why is the RNA primer is in small fragments?
Why in small fragment?
Yes, sir.
Yeah, that's why I told because it is lagging stand because see. First, you have to put in order to make just one, in order to put one primer, like and synthesize, you have to put this primer here, somewhere here. But this part is not available. So you are actually making this fork, like this fork is moving in this direction, like long DNA, right? This portion is not available, only this portion is available. So when this will be like, the position will be something like this, I would say little bit later, say I'm just drawing like, by dotted line, something like here. Then another primer can sit here, like another RNA primer can sit here, because then this is available. But the rest of the portion is still double-stranded helix, so it is not available. So that's why this strand is going into small, the synthesis is happening in small portion, and those. That's why this is called lagging strand. It is going lag behind. This is going slowly. Okay, so that's the reason. So just go through the slide and video and let me like if you have any problem, I will again discuss in the next class. Just ask me some question. So I always do that actually, no problem. So again, I will discuss that in the, like, if you have some specific question, I will address that. But before that, I will request you to go through the slide like carefully as well as the video and then ask me later. And the next thing is very small. This is very simple also, that the replication I told you, but now how this is the date is previous date. So how you can analyze DNA in lab, right? So laboratory methods for analysis of DNA. We are going to discuss about gel electrophoresis, one, and another one is PCR. I told you what is RT-PCR, like one student already told what is PCR, and DNA sequencing. We cannot discuss DNA sequencing today. This is much complicated. I need to spend more time. So how we sequence, how we decode the information present in DNA. Okay, that I will discuss later. But those things will be much more simpler than the replication. What is actually what I discuss in the what is happening inside the cell, because all those complexities should not be there when we are doing it inside the tube. Okay, so first let me talk about little bit about gel electrophoresis. This is very simple. So what we do that we make some kind of gel. Gel means this is agarose gel. So agarose gel means agarose is some kind of I would say compound. Like when say for agarose is some compound, if you boil in water, it will dissolve. And again, if you cool that, like if you decrease the temperature, it will be solidified, right? So then that kind of semi-solid, like soft, but it will be solidified. That makes some kind of matrix. And in that matrix, say for example, this is some kind of gel here. So here. So now, if that solid gel, and there if you load DNA in one end, and that gel you have to, like that semi-solid thing you have to keep in some some buffer, and you have to connect some electrode, and all of you know what is the negative, what is the net charge of DNA? Tell me.
Negative.
Negative.
Right, yeah. So as a result of that, what will happen? Some student actually raised hand. I think you have question. Just give me a few minutes and then you will tell your question. So the thing is that whenever you make this gel, so you have some system like this. So actually the system looks like here. So again, that this is the problem. When I go to the class, I take this system from my lab actually to the class so that you can see actually how it looks. So this is some kind of, just, you have this small system and you put this, you make this gel, this blue color here. This is semi-solid thing. Like now this gel, you put it here and you just immerse the whole tank with some buffer so that electrical conductivity will be better. And now you have negative and positive electrode. So here in this case, for example, this is the negative electrode, this is the positive electrode, and you make some small oil here. You have that opportunity, you can make it actually when you are making the gel, you can do it. So now in the oil,
The DNA is the gel here, or the medium is the gel?
so here DNA is nothing, like I didn't talk about DNA yet. So this is agarose boiled with water. Okay. And when you cool down, that will make this gel. And say for example, like what happened, I know I should mention actually. So when you are making this gel, so you just boil this agarose in water. Like you need to add some salt so that some buffer also, so that electrical conductivity will be better. So you pour that in some some cassette so that you will get some kind of particular thickness of this solid thing when it will cool down. And when it is liquid, you can put some comb. So to make those oil. So comb means just comb. Okay, some like comb are available. So if you put that, and then DNA gets not DNA, the gel gets solidified, and then take out the comb. So as a result of that, in the gel you will get some small oil. So that is the those oils are here. See, those are the sample oil. So you can load sample here, right? So now you have to put your DNA here. You have to load DNA here, and the whole thing you have to place in this box, in this system. So where you filled with buffer also, and now if you connect this with some power supply with negative terminal here and positive terminal here, as you know that DNA is negatively charged, then what will happen? DNA will start migrating from negative to positive direction. DNA will start migrating like this. Okay. Then the composition of gel is same everywhere. So then how you will what will be the determining factor of their migration? Their length of DNA, mass of DNA, because all DNA is negatively charged also. So in this case, you can see in this, like you have 4 oil here, oil number 1, 2, 3, 4, like 4 DNA sample you have loaded, and they have migrated in this direction. Why? Because negative electrode, this is positive electrode. That's why they have migrated. So from this migration, you can say the DNA loaded in the fourth lane, that is the lowest molecular weight, or I would say the minimum length. And this one in the second lane here, you can see this one, this is the biggest DNA, okay, out of these four, because it migrated minimum distance. That means it is much bigger DNA. That's why it is going slowly. So until now, like, if you have any question, tell me. This is very routine work. Every
Sir,
day
I
we
have a
do in
question.
lab. Yeah.
Okay, you go.
Sir,
So
a
what the
bigger
charge
DNA.
contribute here?
So one by one, please. So I can see that Soham Chakraborty actually now unmuted. So can you start, Soham?
Sir, a bigger mass of DNA, except having a greater mass, will also have a greater amount of negative charge, right? So why won't it, why won't these two factors, I mean, how do we decide which would be the greater?
I understand your point. It's like DNA is completely negative charge, right? So I understand your point. But here the gel we prepared, there is already solid, like gel that has some pore size also, right? So as a result of that, although you have negative, like, those are negatively charged, but here, like all, like, like I would say, if you have 100 nucleotide long, you have 100 negative charge, 200 nucleotide, 200 negative charge, for example, I'm saying. So charges all are negative here, but whenever the DNA is much bigger, that cannot actually go through the gel. That's why. So we are actually ignoring here charge, that we are only considering all are negatively charged. But the difference, what you can see based on their like length, I would say mass. But if you say, for example, I'm telling you, so if you do the experiment, so you loaded like 1000 nucleotide long DNA. So if that is true that you have more charge, so it will go faster. But because of this gel matrix, it will not even enter the gel. Like it is too big to go, right? It cannot actually cannot be separated. The only thing what we do, sometimes we actually decrease and increase the concentration of agarose, that agarose gel I told, to make that gel, that how it should be solid. It will be too solid or very, like, less solid, or like that way you can control. But here we are separating based on their like mass or charge. Sorry, not charge, mass or length, not the charge. Because otherwise, if you put like 10,000 nucleotide long DNA, and I am expecting that it will go, but it will not even enter, right? Because of this pore size in the matrix, you have like some porous thing, right? So it will go through, but it cannot. It cannot go because of its dimension. Then it is done, right? So that's the thing.
So this DNA that are being poured into the wells, they are broken, like they are uncoiled, or they are supercoiled, like.
Very
Uh,
good. Very good question, actually. So all are possible. Here, I'm not going into discuss like, like all sort of these things. So for example, it is just imagine that this is double-stranded DNA, and also it forms helix. But there are other techniques where you can, separate that into single strand, and then you can run. That is called a defined types of gel. But intentionally, I'm not bringing those stuff. Just in this case, you think that you synthesize DNA in lab, like in some tube, and then you are analyzing that DNA. So that way you are doing. So it is double stranded, and it is a helix actually. So even the bigger question is, this band, this color you can see, how we will get this band, how you can see that thing, right? Because DNA is, you cannot see in your naked eye, like you know the dimension of DNA. Even one cell you cannot see by your naked eye, apart from some egg are also like single cell. So how you
so
can
we
see
have
this?
some UV equipment in there,
Yeah, very good.
like
Yeah,
that.
yeah, something like that. So what happened? DNA is double-stranded helix. So during this reaction, what we add some dye, some reagent, that is called ethidium bromide. Okay, ethidium bromide. So DNA is double-stranded and is from helix, and nucleotide bases are stacked inside the helix. So ethidium bromide that get intercalated in between those base stacking. So ethidium bromide get inserted inside the DNA. Now, ethidium bromide can fluoresce at certain wavelength. So after running the gel, so if you see this gel by your naked eye, nothing you will get. So you have to put this gel on some UV trans illuminator. That means you have some small equipment where you are providing some UV light on the gel, right? So the wavelength is approximately, normally we use in between 300 to 350 nanometer. That is approximate range I am saying, 300 to 350 nanometer wavelength. Now, in this wavelength, that ethidium bromide, which is intercalated in between DNA, inside the DNA, that fluoresce. That means that absorb the light and emit, and the emitted light or the wavelength is much bigger, around 500. So that's why you can see this kind of little bit orange to red color. See, we provided UV light, but ethidium bromide, when intercalated in DNA, then it actually emit this wavelength around 500 nanometers. So that's why you can see this color. Always this looks like orange to red color, right? So that is how we observe DNA in lab in a routine basis, like the length of DNA. So now you can, is it clear?
So what is the use of that experiment only to determine the length?
Yeah, so the thing is, say for example, it's a good question. So for example, I told you are working in my lab. I told you, okay, synthesize this DNA. So I provided you template. I provided you, all the nucleotides, everything. So now, after the experiment, you did the experiment, the polymerase chain reaction, I will discuss. But after the experiment, how you will determine whether you synthesize the DNA or not? Like if you synthesize the DNA, that means you should have, like, that you should increase the amount, right? So you have, like, initially I have provided you like 10 template or 10 parent strand of DNA. So then if you can amplify it, so see the polymerase chain reaction, I will discuss that later. So you can exponentially synthesize DNA, but then you have to check whether you have really synthesized it or not. So that is the, like, one common technique, that how you can actually see DNA very quickly, within hour, actually within 1 hour you can finish the experiment. And say for example, another thing I'm telling you, in forensic lab, say from some crime scene, like somebody, like some police officer, some forensic team, they collected, like, say, one blood drop, like, or few hair. So even from that sample, some biological sample, you have to extract little bit of DNA. Now that DNA, if you load in DNA gel, you cannot see, because that is too little amount of DNA. So now that DNA need to be amplified. You have to make more DNA out of that small amount of DNA which has been recovered from some crime scene. Now you carry out that polymerase chain reaction. That means inside the tube you amplified that. Then again you have to actually test that whether it has been amplified or not by this gel electrophoresis technique. Very simple technique, but you have to do this. And
Excuse me, sir.
yeah.
Sir, why don't we use centrifugation technique instead of gel electrophoresis to
Yeah,
confirm
centrifugation. Yeah, I understand. But Rajesh, centrifugation technique, it is much longer. You have to load huge amount of DNA in order to see that. Okay, so we never use centrifuge for a routine basis to see DNA. So the experiment I told you, that is long time back. That time neither like gel running system was discovered or nothing, right? So that's why they did in that way. But you have to load huge amount of DNA. But here you need just few nanogram of DNA, just nanogram. Okay, so this is more powerful technique. That's why. But if you have huge amount of DNA, you can do that. That is possible, right? But the thing is, again, you cannot determine the length and so many things. It will be much difficult experiment. But now this gel electrophoresis is one of, I would say, the easiest experiment you can run very quickly, like, and you can, do this thing. So now what I was telling actually, that from the crime scene you collected the sample. So now you actually amplified it and you checked the length of this DNA through agarose gel. and then you have to sequence it in order to like find out who is the who is involved in the crime, and to investigate that you have to sequence to identify the person from where this sample came, right? That is called DNA sequencing. So both of these, like PCR, like polymerase chain reaction, as well as DNA sequencing, I will discuss during next class. Okay.
Sir, after replication, will the property of DNA will change?
Property means what kind of property? Tell me, because it will be. I would say, like, if I want to add, like, answer just in one sentence, I would say
Like
no. Yeah.
sequence of passage, like.
No, sequencing will never change. Otherwise, everything will be messed up. If sequence get changed, then everything will be messed up, right? That cannot happen. That's why this application is so precisely happening, right? A lot of things involved here. That cannot, happen. It should be exactly same copy of DNA. Okay.
Okay, sir.
Yeah. So now I will send you actually assignment, because assignment means not like mandatory, but if you participate, that will be helpful for you in during your exam. So because last week I discussed only few things, but now I will be able to send you like some question so that you can answer those things by your own, or you can send me through email. I will provide you another email ID. So through ERP email, and then one of my TA will check it and provide proper response also. Even sometimes some of the response I also check by myself, and I will provide you my feedback. Okay, that's all. So today it's done. Thank you very much.
Thank you,
Thank
sir.
you, sir.
Yeah.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you,
Thank
sir.
you, sir.